So what you get are huge multiplets that interlace all over the place, and you get an awful mess. So we take the hit and lose that information deliberately by erasing it all. So in a carbon spectrum, you should only see singlets, no doublets or triplets, and you should see a singlet for every chemically different type of carbon. They should also be in approximately the correct regions. And in terms of areas underneath the peaks, we do not use that data. A fully proton decoupled carbon spectrum has areas that are distorted by a technique called the nuclear Overhauser effect, or the NOE. The NOE causes carbons that have attached hydrogens to appear larger in size than they should be. For instance, let's see if I can pick it out here. Well, it's hard to see this one. But you notice the CH2 at 25 is actually taller than the CH3. That's a distortion caused by this NOE effect, the NO effect, the nuclear Overhauser effect. So the bottom line is you cannot get accurate integration data on a carbon spectrum. Now, before I leave this NOE, before you roll your eyes and say, oh, stop talking about those esoteric effects, the NOE is what allows biochemists and molecular biologists to do protein 3D structure by NMR. So at some point, if you stay as real scientists in biochemistry or molecular biology or medicine, you will deal with the nuclear Overhauser effect, because it can actually be used to tell how far two hydrogens are from each other. It's a through space effect that varies with distance. So if you have a big protein that folds, you can have a proton on amino acid 29 that is right next to a hydrogen on an amino acid number 568. Even though the distance is far apart through the bonds, because of the folding, they approach one another within a certain number of angstroms, and you can read that all the way up the protein chain, and you can tell how that protein is folded in 3 dimensions. So you get 3-dimensional data. It's a very complex experiment, but very, very powerful. Before I leave this, you should know that, and I mentioned that MRI is just an offshoot of NMR. Now, in MRI, to go for magnetic resonance imaging, what they have is a regular NMR spectrometer, but it's tuned to water. So it only looks at water in the tissue. The hydrogens of the water in the tissue are affected by their environment. So if you have a hydrogen that is in a cancerous tissue, for instance, in water that's in a cancerous cell, that's different than hydrogen on water that's in a benign cell or non-malignant cell, etc. And so by measuring properties of those water hydrogens, and what they actually measure is what's called the T1 value, which is the time that it takes for a flip to come back down to its original state. It's called the relaxation time. And how long that takes depends on the chemical environment around the water molecule. So again, you get different T1s for different kinds of water molecules that are all the same as water molecules, but they all have a different environment. And that's translated into shades of gray. So a large T1 has a certain shade of gray, a smaller T1 has a lighter shade of gray. And so the computer sorts that out into a slice image. And after taking lots of slices simply by moving the radio frequency transmitter in small increments. So it takes slices through the body part of the whole body. You can get a three-dimensional image. And there are lots of other experiments like spin echo and T2 that you can get done on universities that have, or university hospitals that do a lot of research into MRI. Like UMD&J does a tremendous amount of not only NMR, but also magnetic resonance imaging using techniques that aren't available if you go to Montclair Radiology, for instance. You can only get one kind of experiment. There's a lot that can be done, but it's just an offshoot of NMR. Okay, work some spectra, and we'll do a little bit with this on Thursday, and then we'll be all set.